All right, let's get these muscle contractions started today. So that's essentially what we're going to be going over in this video is just muscle contractions and how that works at a molecular level. So I'm going to begin by talking about the sarcomere, which is the functional unit of muscle. It's the basic contractile unit of the muscle cell, and I'm going to draw here a Z disc. Which these Z discs are the ends of a sarcomere. So essentially you have many sarcomeres lined up together to make up your muscle fiber, and then on the ends of them you have Z discs in between. So you have your sarcomere here and Z disc here. So that's what I'm going to draw right now, the Z disc. And this is where action happens. Let's see. So again, you have multiple of these along a single muscle fiber. And I'm going to draw like this. I'll explain here in just a moment what this is. Alright, so here, these straight looking rods are nebulin. And this is where we're going to have our actin. And then here we have our titin, which has a spring-like structure on the ends, which allows this portion of the titin here, which has the myosin, to move back and forth as the actin, where the nebulin is here, slides over it, kind of like if you were to slide a ladder over itself. That's how that would function. So now that we see what a Z disc looks like and the components of it, I'm gonna just draw here some actin. So each one of these little circles here is what we would call G-actin. And when you combine multiple of those together, you get F-actin. And then here on the titin, we have myosin. And that's the same here on both sides here as well. It just continues on. But I'm just going to focus. I'm just going to draw it once so we can see what it looks like. No need to draw it more than that. So something that's important for us to understand is that this F-actin, I'm going to draw it right here again, a little bit bigger, has all these G-actin proteins connected together, and they're also going to have a protein called tropomyosin that I'm going to symbolize as this kind of wavy line. I guess I better make that a little bit longer. that's going to block these sites on the F-actin, or these G-actin proteins, where the myosin heads are going to bind. So again, you have your sites here, and these myosin heads are going to bind there, like so. This tropomyosin, however, is a problem, because if our myosin down here wants to bind to these sites, the tropomyosin's in the way, and so we need to remove that. And that is done with another protein here called troponin that has 3 components, as you can see. So I'm going to draw it over here like this, a little bit bigger. So you have your troponin I, troponin C, and troponin, let me think for a second. I believe it's T. And so troponin I is binding to the G-actin. Troponin T is binding to the tropomyosin. Troponin C binding to calcium. Calcium is super important because calcium drives muscle contractions, as we'll get into here in a moment. Something else to note is that this calcium has 4 binding sites. 2 of them are low affinity for calcium, and 2 of them are high affinity for calcium. Now, back to this image here. So when we have high amounts of calcium, we already have calcium that's bound to these high affinity sites, okay, right here and right here. And now when we have high amounts of calcium, we also have calcium bound to these low affinity sites. And that's important because in order for troponin to remove this tropomyosin, making these binding sites on the F-actin available for the myosin to bind, we need calcium on these low affinity sites. So when that happens, troponin pulls this tropomyosin out of the way, and these myosin heads are able to bind. So it looks something like this. This would get moved out of the way. And now these myosin heads can bind to the F-actin. Now, let's we're gonna look. You might ask, well, why is any of this important? Again, keeping in mind this little ladder model, this myosin is going to bend or move this way, and it's going to pull the F-actin over itself. Now, you don't have all the myosin heads binding at once, but you have them alternating, or some binding at one moment, some at the next moment. So you continuously have this movement of, this actin, for example, going this way, and myosin coming this way to shorten the muscle. So we're going to talk a little bit more about what that looks like, this process here, next, when we talk about muscle contraction. And then I'm going to look at the bigger picture, and we're going to look at action potentials and how they propagate from a neuron to the muscle fibers to cause this contraction. So again, looking at this myosin and F-actin, how does that work? Well, let's take a look. So this line here is going to represent our F-actin, and this is going to be a single myosin head, and this kind of works in a little bit of a cycle. Now, this is going to be specifically skeletal muscle contractions, and I'm just going to draw this out. Again, here's our F-actin and our myosin. Make sure we have enough room here. Might need to shrink that, but we'll talk about it first. All right. So as you can see, this is a cycle, it repeats over and over again. This is how, looking more at the kind of molecular level of how this F-actin and myosin interacts, like we were discussing earlier. So, begin here. Let's see. With calcium, right? So we have this calcium coming into the cell, the muscle cell. And again, we're going to look at how these high amounts of calcium occur in the muscle cell in a moment. But just for now, all you need to know is that this calcium is coming in to the muscle cell, so it's going to be a part of our cycle here. And remember, this calcium is going to bind to troponin. Troponin will remove tropomyosin, and that'll make the binding sites on the F-actin available to the myosin head. And so that's going to cause, when we have this calcium coming in, that's going to cause this right here. Notice this difference. Myosin head is now bound to the F-actin, which is going to release heat. And now our muscles are engaged. And at this point here, well, actually, we start here with ADP and a phosphate there. We have calcium come in, and we still have this ADP and our phosphate, but now the myosin is bound to the actin. And when that happens, when this binding happens, not only does it release heat, but as we see in this next step, it's also going to release the ADP and the phosphate, leaving us this phase where when this ADP and this phosphate come off, it's going to cause this myosin to go from a cocked position here to an uncocked position here. As you can imagine, that's pulling this F-actin like this. And then we're going to have ATP that's available in the muscle cell that is going to bind to this myosin. And when that happens, notice that the myosin is going to be released from the F-actin. And when that myosin is released, we have hydrolysis. Where we're going to lyse or remove a phosphate from the ATP and end up here with ADP and phosphate. And notice that this will also cause the myosin to cock. Go back into the cock position like shown here. And then that cycle will repeat itself. As long as we have calcium, that is indirectly causing the availability of the F-actin binding sites for the myosin. So that is skeletal muscle. Let me see if I can shrink this a little bit. Make some room. There we go. I like it. Okay. Now, that's kind of at the teeny tiny level, right? More at a kind of microscopic view. Now I want to take a big picture view for a moment and look at action potentials, how they propagate and cause any of this to happen in the first place. So let's kind of take a bird's eye view here now. I'm going to draw, let's see, I'm going to use blue. No, I'll just use black for this. I'm going to use black to draw right here our neuron, our postganglionic neuron here, coming down, and yeah, I'll just do this. And here's our terminal end. And let's see, here's our muscle cell, our muscle fiber. Right, come in here. We have our synaptic cleft here. And then we have right here what we call a T-tubule, which is just like a little narrow tunnel into the muscle cell fiber, or the muscle cell. But we still would consider this. Here's our ECF, extracellular fluid, same here. And then ICF, here's our intracellular fluid. All right. So on our neuron, we have several channels. We also have myelin on the outside. And we have some channels and voltage gated channels. So for example, here we have a potassium leak channel here, and we'll put our voltage gated sodium channel. We have those. We have some voltage gated calcium channels, and we also have these vesicles here in the terminal end of the neuron. And these vesicles have, make this green, they have acetylcholine hanging out inside them. the neurotransmitter. They also have, let's make this red, these snare proteins, right here. And what happens is we have our action potential that's propagating down the neuron all the way down here to the terminal end. And that's going to cause our voltage gated calcium channel to open, which is really important because now we have all this calcium that's coming into the cell. And this calcium will interact with these snare proteins, which will pull the vesicle up to the edge right here of this terminal end of the neuron, and will, whoops, not what I wanted, will cause I just have to erase it this way, will cause the release of acetylcholine into the synaptic cleft. Why is that important? Glad you asked. It's because we have these, I guess we'll drop in blue. We have these nicotinic 1, and I know I'm not drawing like the gate where it's closing and everything, but it's just quicker and easier to draw the channels this way. I guess I could like use one example here where the gate's open like this because we have acetylcholine acting as a ligand binding to these nicotinic 1 receptors, and that, my friends, when these gates are opening, we have sodium rushing into the cell. And now, the sodium that's coming into the cell is going to cause our wongs, as Brother Shaw would say, which further propagates our action potential along the membrane all the way down, 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 as we reach the T-tubule and come down this way. And I'm just going to kind of draw a block here to represent what we call our sarcoplasmic reticulum, or I'm just going to abbreviate it as SR. Now, as this action potential propagates along the cell membrane, we have a type of calcium voltage gated channel called DHP. That's in the cell membrane. And then in the sarcoplasmic reticulum membrane, we have, I should have put these a little closer together probably. We have another channel that's, let me see what color. I guess I'll just do blue. Another channel here called our ryanodine channel, so abbreviate that as RYR. And what happens is this action potential causes this DHP to open, but when it opens, it can't open fully and pushes open the ryanodine channel. And now we have all this calcium that's being generated inside the sarcoplasmic reticulum, which, by the way, is where we're getting most of our calcium from inside the cell rather than from the ECF. We have all this calcium coming out of the ryanodine channel into the sarcoplasm. Why does that matter? Well, remember what we talked about earlier with the Z discs in the sarcomeres. When we have this calcium, we're able to have muscle contractions now because we have this myosin that's able to bind to the actin. So that's why it matters that all this calcium is coming out of the sarcoplasmic reticulum. And that calcium will be used by the sarcomeres. Now, we also have what we call here, I'm just going to use arrows for these channels, or for this pump, that's known as the Surca pump. And the Surca pump is going to take the calcium that's building up outside of the sarcoplasmic reticulum in the sarcoplasm, and it's going to pump it back into the sarcoplasm, therefore recycling this calcium. And that is how a normal propagation of an action potential would occur for muscle contractions. So, we've talked about the sarcomeres, we've talked about action potentials in the sarcolemma and muscle contractions of skeletal muscle and how that looks. So now it's time for us to move on to talking about energy sources of muscle contraction. So with energy sources for muscle contraction, we have different different levels, if you will. So the first one is going to be cytoplasmic ATP. This energy source is used very quickly. It only lasts for about 5 seconds of a contraction. And then the next energy source that would be used after that, from between 5 seconds and 10 seconds, is our creatine. I want to say creatine monohydrate, but I think that's the supplement. Let me think for a second. Creatine phosphate. That energy sourced, it will be used for the first 10 seconds, for between 5 and 10 seconds. So these are quick muscle contractions. I believe, when we have eye movement, for example, that's a brief muscle contraction. Or if we were to pick something up off a desk, that could be these short, muscle contractions that would use cytoplasmic ATP and creatine phosphate. But as we know, sometimes we need to engage our muscles for longer and have longer contractions. So we have 2 other energy sources we'll talk about. The third one is glycolysis from glycogen stores in the muscle. And that will get us to about 60 seconds. So a whole minute. Oh my goodness. So you're carrying groceries up a flight of stairs. There you go. You're probably hacking into that glycolysis. And these are all anaerobic. I don't think I spelled that right. Anaerobic sources of energy for muscle contraction. And then the fourth one, because you're probably thinking, well, I've done wall sits for 5 minutes because you're an absolute beast. That's where we're getting our aerobic respiration. And that'll carry us through for hours. So that would be the energy source used to run 10 miles, for example. Or if you're in the gym lifting weights for any period of time more than 60 seconds, it's likely that your body's going through aerobic respiration as an energy source. Okay, I probably need to plug my tablet in because it's running low on battery. So, let's see. Alright, that's better. Okay. So that's energy sources for muscle contraction. Now, so far, we've primarily been talking about skeletal muscle. But what about the interactions of myosin and actin and smooth muscle? Smooth muscle, for example, in organs that contract involuntarily without our conscious effort, or smooth muscles found in sphincters throughout the body, etc., that can contract for much longer than we can hold a contraction with skeletal muscle. Sorry for all the yawning, by the way. It's been a long day. All right. So there's two kinds of smooth muscle contractions. And I'm going to draw this. It's going to look at first very similar to skeletal muscle, but there are some key differences that I will point out. So feel free to skip past all this drawing. Oh, goodness gracious. Some might even say I could do this in my sleep because I'm yawning so much. No, not really. Solo bromas. Solo bromas. I think I'm following Brother Hunt's pattern of just caring less and less what I say about the content during a lecture video. Just getting goofy. Okay. Let me copy this. Because, like I said, we have two of these. Two different kinds. The first one we'll talk about is what we call phasic. And the next one is what we refer to as tonic. So first, we'll talk about phasic. Now, phasic, again, calcium. We need calcium. That's our starting point. And with this smooth muscle contraction, something to note that's a little bit different here with the calcium is that calcium, instead of being used to bind to troponin, calcium affects our myosin light chain kinase, which is an enzyme involved in smooth muscle contractions. And I will highlight that there is no troponin involved with smooth muscle contractions. So again, calcium is not going to interact with the troponin because there is none here. Instead, it's going to be interacting with MLCK, which I will actually draw out here in just a second. But before I do that, I just realized I also failed to mention earlier that these cycles that we're looking at are what we call or refer to as a cross-bridge cycle. Just wanted to point that out before I forget. So I'm just going to come up here to kind of like try to visualize MLCK. We're going to draw this enzyme and make it look like a snake. All right. So this is our MLCK. And there's a protein called calmodulin. I don't know if I spelled that right, but it has binding sites for calcium. And so when we're talking about this calcium here, right, we have calcium that is binding to this calmodulin. That causes calmodulin to come over here and bind to this MLCK. And when that happens, this MLCK undergoes a, I can't think of the word right now, but basically a structural, or a conformation change. And we pointed out that kind of looks like a snake earlier, which whether it does or doesn't, I don't really know. But point is that now this MLCK has undergone a conformational change, which is going to activate it, and it's going to use ATP to add a phosphate to this myosin. So a kinase is going to add phosphates, right? So we have our MLCK, myosin light chain kinase, and it's going to use ATP to come over here, and we have our ADP here and a phosphate, just like in skeletal muscle. But now, okay, because it's MLCK, got a phosphate, an additional phosphate here. And as normal, this myosin is going to bind here to the actin. And just like in our skeletal muscle, when that happens, ADP is actually going to fall off. And we'll have a phosphate come off as well. But we'll retain this phosphate here. So as usual, myosin goes from a cocked to an uncocked position, pulling that F-actin. Now we've got ATP that's going to detach the myosin from the actin, like so. And now this cycle can take two paths. Either it can continue on where we have MLCP, so this is a myosin light chain phosphatase, and the phosphatase, that enzyme has the job of removing the phosphate. And that enzyme is influenced by Rho kinase. And so when you have high amounts of calcium, you have low amounts of Rho kinase and low amounts of rho kinase means active MLCP, which is going to snip off that phosphate, and you'll end up here with your myosin. However, if you have low amounts of calcium, you have high amounts of rho kinase, and that is going to inactivate your MLCP, and that allows us to keep that phosphate and undergo hydrolysis, which will bring us here, just right back here. So we kind of skipped a step above. I think I may have left a phosphate off there. So that is our phasic cross-bridge cycle explained to you. Now we're going to look at tonic. So tonic, again, still smooth muscle, but this is going to be slightly different. So we're still going to have our calcium coming in here. We're still going to have our MLCK, and that's going to be using ATP. To add this phosphate here. But now the difference is that with the tonic cross-bridge cycle, we still lose this ADP comes off and phosphate. But now here we have MLCP that's going to be active. And it's going to, remember, it's phosphatase, so it's going to remove phosphate. So now we have just this bare myosin that has been uncocked. And as usual, ATP is going to try to come in here. But because we don't have that phosphate here with the smooth muscle like we did previously, we have this ATP that's trying to bind here to the myosin. But it's having a hard time. Keeps trying, keeps getting denied. And so because of that, this actually takes a longer time to get back to this phase here where we have ADP and a phosphate, and we call this the latch part of the tonic cross-bridge cycle. So those are the two cross-bridge cycles that are involved with smooth muscle contractions, and you can see how they're different from skeletal muscle contractions. But the advantage of smooth muscle, or I guess the different function, is that these contractions can be much, much longer than that of skeletal muscle because of these differences. So I just talked about the difference between smooth and skeletal muscle. So we'll just move on to muscle spindles and Golgi tendon organs. And you're probably excited because you know that means we're almost done, and you're probably tired of hearing my voice. So let's go ahead and jump in, shall we? So if we were to draw some muscle fibers here, we would find that we have what we call these muscle spindles that run in between these muscle fibers. So you have this comes like this. They're squished in there. And they don't run along the entire length of the muscle fiber. but there's several of them that are along the muscle fibers, right? But the spindle itself doesn't run the entire length. There's just multiple of them. And in this muscle fiber we have, or in the spindle we have, let me draw this like this. Oh, that doesn't look very good, but we'll, oh yeah. We can make do with what we got because we are resourceful. All right. Let's see. We have I believe on the outside here, what's referred to as extra fusel fibers, if I'm not mistaken. And if I am, I'm sorry. And then here on the inside, we have intra fusel fibers. And we have two types of intrafusal fibers. You'll notice like these ones that are running here along the middle. Not necessarily that's their orientation, but these are the chain fibers. And then these bulging looking ones on the inside, those are our bag fibers. And I'm going to kind of like, let's see, just draw them like this. So here's a bag fiber, here's a chain fiber. Got both of these in your muscle spindles that are tucked away between muscle fibers. They're used for reflex. Okay? Reflex. I'll talk a little bit more about that in a second, but to continue on with the structure of these, right, we have our kind of like our sarcomeres here. But not here in the middle of the bag fiber. And then our chain fiber here. And here we do have sarcomeres that are running through the entirety. But on both of them, we should note that we have these neurons that innervate or wrap around these bag fibers, and they send a signal to the spinal cord. And I believe the same here for these chain fibers. Oh. Now these ones will be, the bag fibers will be type 1A, just means that they're fast. Responsive, quick. And for chain, I believe they're 2A, which means simply they're just not as fast. Not that they're not fast at all, they're just not as fast as the 1A type fibers. Now because these ones are fast, they can adapt quickly, and they're more rate oriented. when they respond to stimulus, which that stimulus would be stretch in this case. And then for chain fibers, this is more for duration oriented. Not as fast, but can go longer. So, let's see. Now, I remember Brother Hunt explaining or talking about gamma neurons innervating intrafusal and alpha neurons innervating extrafusal, and their response to stimulus. Honestly, that's about the extent of what I remember. I'm having a hard time with the recall on that right now, but when we kind of zoom out in a big picture and look at this, like why this even matters at all, I'm going to draw spinal cord here. So this would be the back of the spinal cord, the front, dorsal and ventral. And let's say we have a leg here, right? Here's our tendon, where our knee is, and now we've got quadricep, a hamstring. We're gonna have our motor neurons come back in here through the dorsal horn of the spinal cord, and this is going to be sending a signal to the spinal cord. If we have this muscle and it's stretching, the actual muscle fibers, right? Or I should say these muscle spindles, if they're stretching, that's going to send a signal to our spinal cord through the dorsal horn. And then we'll have so it'd be sensory neuron, right? Then our motor neurons, right? We'll have one that'll come back to the quadricep. We have one here going to hamstring. And if sensory information is sent of the quadricep stretching because of those muscle spindles, then what we have here is, let's see, actually, so I believe this would come, let's see, let me think for a second. I believe this is going to synapse here on this motor neuron that's going to the same muscle. And then we have something, neuron branching off here, that's going to synapse with an interneuron that will then synapse with the motor neuron. And what happens is you have an EPSP because the brain is, or the spinal cord is sensing, right, of the stretching of the muscle. This reflex is the motor neuron is going to come back here and it's going to contract the muscle. And this other neuron here is also saying an EPSP to contract this muscle, but because of this interneuron, it has now switched it to an IPSP. So it's going to inhibit or prevent the hamstring from contracting, so it'll relax. And that's just to help protect our muscles. So that's a stretch reflex there. Okay, now our Golgi tendon organs, abbreviated as GTO. Gonna work a little bit different. So I'm just gonna copy this here. So I don't need to draw that all again. Our Golgi tendon organs, instead of the sensory information coming directly from the muscle spindles in between the muscle fibers, it's actually going to come from the tendon. So when there's tension coming from the tendon, because we're contracting our muscles, contracting them, that's causing this tension in the tendon. Tension in the tendon. That's sending sensory information to the spinal cord. Oh, and now we're going to kind of flip-flop this where we have an interneuron here going back to the same muscle, and that is going to cause ultimately an IPSP here for the muscle that's contracting, and an EPSP for the antagonistic muscle. So if you have all this tension in the tendon, this, let's say your quadricep is contracting, then it's going to send this information, resulting in IPSP, which is going to, your muscle is going to try to relax so it doesn't overcontract and damage your muscle or your bones. And then this other neuron that's branched off here is going to cause an EPSP, which is going to actually contract the hamstring or the antagonistic muscle to prevent overcontraction of the agonist muscle. So that's a little bit on muscle spindles and Golgi tendon organs. Maybe I need to review that to go a little bit more in depth myself and gain even better understanding, but hopefully this was understandable. Thanks for taking the time to look over this. If you have any feedback for me, I'd love to hear it. And I hope you have a blessed day.